Hey, what's going on? It is me. It is a new day, Wednesday, hump day, January 12th, 2022. And the immune system knowledge and learnings continue to move forward. Um, so, yeah, as always, there's a lot to learn. And today, we're sticking at a higher level of understanding of a function of the immune system and where it comes into play, using some of the concepts we learned at the lower level. Okay, so specifically, we're going to be talking about grafting today, organ and tissue grafting. Okay, and just at a very high level, it's a very complicated science, of course, and it's a very fundamental science to human survival. And the immune system plays a very important role in the successful transplantation of tissues and organs. Okay, so basically, what happens? What is a tissue? What is an organ transplant slash graft consist of? So, if you have an injury, right, and the tissue that's injured needs to be replaced, or the entire organ that's damaged needs to be replaced, you can do so, and this is basic knowledge for most people, but you can do so by having either tissue replaced from your own body and transferred to the site of damage. In certain cases, that can be done, like in the case of skin cells. Or you can have an organ transplanted from another individual to replace your damaged organ. Okay, so in the case of self-transplantation of tissue from one site on your body to another site, that's called autografting, right? Autograph, like a picture, like a signature, but it's an autograft. Okay, the transplantation of tissue from another donor individual, or an organ from another donor individual to your body, is called allografting. And allo means different. Okay, auto means same, allo means different. So, what is the issue with that? Why can't I just take an organ from, like a heart or a lung from somebody else, and transplant it into my own body and have it function normally? Well, on a very high and basic level, the reason is because your immune system sees these foreign cells as invaders, right? It treats these foreign cells. It has no idea what these cells are. They've been introduced to your body, and it's from the perspective of your immune system, the organ slash tissue looks like a massive pathogen, or and it's got its own antigens on it. It's got its own identifying chemicals, and it's got its own activity. And your immune system, in trying to protect you, will attack these organs through the tip and these grafts if there's no compatibility there, and it will do so through the techniques that your immune system has. We've discussed that it has used to attack other pathogens that are legit pathogens, like single-celled bacteria or parasites or whatnot, or the cells from somewhere else that are invading your system. So it'll use cytotoxic T cells. It will use antibodies. It'll go through a plethora of activity. The cytotoxic T cells will just try to destroy the host cells through porphyrin activity, right? Your complement system may come into play as antibodies and complement C3 opsonize some of the cells and then label them for attack by macrophages in case that they're small enough, or attack from killer cells, killer T cells. And that's it for that organ. The organ is going to be slowly destroyed by your immune system, or quickly destroyed, and thus it won't even function for you, or it'll function for some small amount of time in its intent. For example, the heart will pump blood until the heart is damaged irreparably. The transplanted heart is damaged irreparably by your immune system, and presumably you die. So how can we prevent this from happening, and why does it happen? So the immune system essentially, right, the immune system, the cells of the immune system, they how do they recognize? They need to recognize your the transplanted donor cells, right? So how do they recognize these donor cells? So it's all through antigen recognition. Remember the major histocompatibility complex, right? The MHC, MHC is major histocompatibility complex is the primary recognition mechanism of the cell. And remember, it's of immune cells. Immune cells communicate with each other through MHC class 2 molecules, right, that are embedded on the surface of the cell. And there's two, and we also have MHC class 1 molecules. Okay, so the MHC class 2 and MHC class 1. MHC class 1s are what T cells use, CD8 positive cells use to determine whether they're going to kill them or not through the antigen that's presented on these MHC class 1 cells. Okay, so remember, you have self-antigens on your body, right? You have your own antigens that bind to these MHC complexes. And if your cells express these antigens and a T cell happens to come by and check through its T cell receptor, check out this MHC class 1 and see that a self-antigen is bound to it, then it'll leave it alone. But if the T cell sees that the antigen that's bound to this MHC class 1 is not recognized, right, is not a self-antigen, or if a self-antigen won't bind to this MHC class 1, this specifically this groove area, right, this antigen binding groove, then the T cell will think this is an invader and attack it. So your body, this antigens your body produces are actually called human leukocyte antigens, HLA. Okay, it's a very big topic, HLA. But human leukocyte antigen are genes in the major histocompatibility complexes that help code for proteins that differentiate between self and non-self. So the human leukocyte, the MHC class 1 molecules, right? This is kind of like the genetic map of what's going on here. These MHC class 1 molecules that are embedded, or complexes that are embedded into the phospholipid bilayer of cells, they are differentiated via your genome, as all proteins are, and the specifics of their structure, because when you go down to the actual like molecules and the atoms that are, and the amino acids that comprise these protein chains, these alpha helix protein chains, for instance, you'll see great variation that occurs due to the specifics of someone's genetics, right? So the specific genetics of any of these antigens is essentially all located on certain gene locuses, and they, if we magnify it here, we can kind of see what's going on. But they're co-located on chromosome 6, specifically the small arm of chromosome 6, and there are many of these HLAs, and a person will inherit HLA gene alleles from their parents in the same way that standard inheritance occurs, right? The inheritance that everybody knows and learns in high school from genetics through like Punnett square matching, etc. But there are so many different types of alleles and polymorphisms of these genes that it's actually there's this structure, this MHC class 1 and class 2 structures is quite complicated, and all the complex regions require there to be a lot of different alleles that code for them. The good thing is that people only really care about these 2 peptide binding grooves, because this is where histocompatibility is determined by your immune cells, your T cells, your white blood cells, etc. And so in order to make sure histocompatibility works, we just have to find the genes, someone with the genes that encodes for grooves that are similar to the genes for the MHC grooves that the donor has, or sorry, not the donor, the transplant patient has, right? So as long as the grooves to kind of hold on to the antigens are similar enough, then the body will not cause an autoimmune response, right? So that's typically what people do through HLA tissue typing, okay? Tissue typing is the process by which people kind of look for the HLA markers that are most similar. And your parents really have the HLA markers that you inherit, and you are inheriting half from each parent, haplotypes. That's why it's called a haplotype. But really what's needed, what you have to do is you have to find somebody with similar HLA antigen binding genes and alleles, and in order to do that you're going to have to look for many donors, and there's going to be several tests that occur, including molecular tests to determine whether how close molecularly the antigens are from one person to another, the human leukocyte antigen genes or alleles. And then you're going to have to do other tests like mixing with blood and cross-matching. There's a lot that goes into tissue typing, but let's say it's obviously a full-time job for many people. And it is interesting because, as far as humans we get pretty far, but this is something we still have to deal with. Let's say that tissue typing, you're not finding a compatible organ. There's other ways you can deal in organ transplants with the immune response, or the autoimmune response rather, to that foreign organ. And some of it includes trade-offs like immunosuppression, for instance. And immunosuppression is basically where you tell the immune system to stop. It can occur through several methods, through stopping of signaling on cytokine emission, or it can happen through, and that can itself happen through things that prevent DNA replication and transcription. And this is bad because immunosuppression is good, while it will stop your immune system from recognizing your organs. In many ways, the basic immunosuppression isn't targeted enough to prevent your cells from or to allow your cells to recognize their standard antigens like colds and things like that. So people who are immunosuppressed can succumb to simple diseases like colds or flus, or even cancer, because your immunosuppressed cells stop your natural killer cells, stop recognizing cancer cells, and therefore the cancers can proliferate in your body much more easily than they do when the immune system is functional. So anyway, that's a handful of a lecture. I just wanted to leave you with that, and I hope you get a chance to think about all this stuff, and leave me a like, comment, and subscribe. I'll see you later. Bye.